Namaste, Hamisha Kushro Vidya. I am JV Dr. Meenakshi, Assistant Professor in Department of Shalakya Tantra, Faculty of Ayurvedic Science, JAYOTI VIDYAPEETH Women's University. In the previous session, we studied the applied anatomy of the pathophysiology of the glaucoma. We were studying the principal ocular structures. We have already discussed the ciliary body, the angle of anterior chamber, and then we were studying the aqueous outflow system. It includes trabecular meshwork, Schlemm's canal, collector channels, aqueous veins, and episcleral veins. So we're discussing the trabecular meshwork. It is a sieve-like structure through which aqueous humor leaves the eye, and it consists of three portions: the uveal meshwork, corneoscleral meshwork, and the juxtacanalicular meshwork. In this image, they have shown this blue color sieve-like structure is the uveal meshwork, and red color is the corneoscleral meshwork, and this green color structure is the juxtacanalicular meshwork. So uveal meshwork, it is the innermost part of the trabecular meshwork and extends from the iris root and ciliary body to shell base line. The arrangement of the uveal trabecular bands create openings of about 25 uh, millimeter, um, create openings of about 25 millimeter to micrometer, should be, I think, micrometer to the 75 micrometer, and least resistance to flow. Then corneoscleral meshwork, it forms the larger middle portion which extends from the scleral spur to the lateral wall of the scleral sulcus. It consists of the sheets of trabeculae that are perforated by elliptical openings which are smaller than those in the uveal meshwork. They are from 5 micrometer to the 50 micrometer and moderate resistance to flow. Then third is the juxtacanalicular or endothelial meshwork. It forms the outermost portion of meshwork and consists of a layer of connective tissue lined on either side by the endothelium. This narrow part of trabeculum connects the corneoscleral meshwork with the Schlemm's canal. In fact, the outer endothelial layer of juxtacanalicular meshwork comprises the inner wall of Schlemm's canal. This part of trabecular meshwork mainly offers a normal resistance to aqueous flow. This was the trabecular meshwork. You can see here is the Schlemm's canal. Then the uh, next structure of aqueous outflow system is Schlemm's canal. It is endothelial lined oval channel present circumferentially in the scleral sulcus. It is present circumferentially. You can see this is the Schlemm's canal of green color. It is present circumferentially in the scleral sulcus. The endothelial cells of its inner wall are irregular, spindle-shaped, and contain giant vacuoles. The outer wall of the canal is lined by smooth, flat cells and contains the openings of collector channels. Here. The aqueous humor will flow from the trabecular meshwork for the extracanalicular trabecular meshwork to the Schlemm's canal, then it will go to the collector canal. So next structure is the collector channel. Uh, these are also called the intrascleral aqueous vessels, and they are about 25 to 35 in number and leave the Schlemm's canal at oblique angles to the terminate into episcleral veins in a laminated fashion. These intrascleral aqueous vessels can be divided into two systems. There are two systems, and the larger vessels, which are called the aqueous veins, They run a short intrascleral course and terminate directly into the episcleral vein. This is called the direct system. And the many smaller collector channels form an intrascleral plexus uh, before eventually going into the episcleral veins. It is called the indirect system. Here is this an image. This is the this is the aqueous vein. This is the trabeculum ashwa. From that it will go through the aqueous vein. This is the direct system. And then there is an for the smaller collector channels. There is a intrascleral plexus before going into the episcleral vein. They both will go into the episcleral vein. In the direct system, it will go through the aqueous vein. In the indirect system, it will go through the intrascleral plexus. This is the episcleral vein. Most of the aqueous cells drain into the episcleral veins, which again drain into the anterior ciliary and superior ophthalmic veins, which in turn drain into the cavernous sinus. Then is the applied physiology of the glaucoma. The physiology, it is concerned with the dynamics of aqueous humor, its production, drainage, and maintenance of intraocular pressure. First is the aqueous humor and its production. Um, the volume of aqueous humor in the anterior chamber is 0.25 ml, and in posterior chamber it is 0.06 ml. The aqueous humor is a clear watery fluid filling the anterior chamber and posterior chamber of the eyeball. Then the functions of the aqueous humor are: it maintains a proper intraocular pressure. It plays an important metabolic role by providing substrates and by removing metabolites from the vascular cornea and lens. It maintains the optical transparency, hence has the optical function, and it takes the place of lymph that is absent within the eyeball. It has a clearing function. It clear the metabolites. Then the refractive index of the aqueous humor is 1.336. Then composition or the constituents of the normal aqueous humor are: the maximum portion is water, which is 99.9%, solids are 0.1%, which includes proteins, the colloid content, and because of the blood aqueous humor, the protein content of aqueous humor is much less than that of the plasma. However, in inflammation of uvea, iridocyclitis, the blood aqueous barrier is broken and the protein content of aqueous is increased. Amino acid constituent of aqueous humor is about 5 milligram per kg water, and non-colloidal constituents in millimoles per kg water are glucose, urea, ascorbate, lactic acid, inositol, and potassium ions, sodium ions, chloride ions, bicarbonate ions. Oxygen is present in the aqueous humor in dissolved state. Thus, the composition of aqueous is similar to plasma, except that it has a high concentration of ascorbate, um, pyruvate, and lactate, and low concentration of protein, UV, and glucose. The aqueous humor of anterior chamber is different from the aqueous humor of the posterior chamber. The compositions differ, and the main difference is the the bicarbonate in the posterior chamber aqueous is higher than in the anterior chamber, and the potassium concentration, sorry, chloride concentration in posterior chamber is lower than in the anterior chamber. And also apart from that, the ascorbate concentration of posterior aqueous is slightly higher than that of the aqueous chamber, anterior chamber aqueous, sorry. uh, Thank you so much. This session is powered by JAYOTI Vidyapeeth Women's University, and I hope you are satisfied with my session. If you have any query, please mention in the comment box. I will resolve it. Thank you so much. Which is the one third of the total refractive power of the eye. It is a 3/4 of the total refractive power of eye. That is, the total refractive power of eye is 60 diopter, and that of cornea is 45 diopter. Then the histology, there are 6 layers: anterior epithelium, Bowman's membrane, central stroma, Dua's layer, Descemet's membrane, endothelium. You can see in this image the epithelium, Bowman's layer, stroma, Dua's layer, Descemet's layer, and endothelium. First, the epithelium. The cornea epithelium is stratified, squamous, and non-keratinized. It is continuous with conjunctival epithelium at limbus, but having no goblet cells. Thickness is 50 to 90 micrometer. Consists of 5 or 6 layers of nucleated cells resting on a basal lamina, namely the basal cells, wing cells, and the surface cells. The basal lamina is secreted by the basal cells. It is 0.1 to 1 micrometer wide. Ultrastructurally, it is distinguished in two parts: lamina lucida and lamina densa. Lamina lucida is superficial, lamina densa is deep osmophilic. Then epithelial turnover. The early study suggested that the epithelium replaced approximately weekly by division of basal cells and the older shell from the surface. It is now recognized that the germinative region lies at the limbus. The stem cells and the cells migrate at a very slow rate to the center of cornea, which may be as long as the ear. Second layer, the Bowman's membrane. It binds the corneal stroma anteriorly with basement membrane of epithelium. It is not a true elastic membrane. Once destroyed, does not regenerate. Shows considerable resistance to infection and injury. Third layer is the stroma, which is 0.5 mm thick. The stroma or substantia propria occupies about 90% of the total corneal thickness, composed of corneal collagen fibrils, keratocytes, and extracellular ground substances. The lamellae are arranged in many layers, 200 to 250, and are parallel to each other. The anterior one-third stroma has a lamella are thin, and in the posterior two-third of the stroma, the lamellae are larger. Next layer is the Dua's layer, which was discovered by Dr. Harminder Singh Dua at the University of Nottingham in 2013. It is a fourth layer which is 15 microns thick. It is a strong layer and impervious to air. Fifth layer is the Descemet's membrane. It is a strong homogeneous layer. It is a basement membrane of endothelium made up of the collagen and glycoproteins. It is elastic. It can regenerate. It has a resistance to chemical agents, infection, and pathological processes. The peripheral rim of Descemet's membrane is the internal landmark of the corneal limbus, and also it is the anterior limit of the trabecular measure, which is called the Schwalbe's line. Then the last layer is the endothelium. Corneal endothelium is a simple squamous epithelium. Endothelial cells are hexagonal, which forms a continuous mosaic pattern, best seen in specular microscopy. Cell density at birth is 6,000 cells per micrometer square, and in young adults 2,500 to 3,000 cells per square millimeter. The endothelial cells cannot divide or replicate. With aging, the cell density of the endothelium decreases, which is compensated by an increase in the cell size or shape. As these endothelial cells are involved in corneal hydration, they help in maintenance of corneal transparency. The blood supply of cornea. The cornea is a avascular structure. Small loops derived from the anterior ciliary vessels invade its periphery for about 1 mm. Actually, these loops are not in cornea, but in the subconjunctival tissue which overlaps the cornea. Then the nerve supply of the cornea. Cornea is supplied by the anterior ciliary nerves, which are branches of the ophthalmic division of fifth cranial nerve. After going about 2 mm in cornea, the nerve loses their myelin sheath and divide dichotomously and forms three plexus: stromal plexus, subepithelial plexus, and the intraepithelial plexus. Then the applied physiology of cornea. Primary physiological functions of the cornea are to act as a major refractive medium and to protect the intraocular contents.
Then the last point is corneal transparency. It is a result of peculiar arrangement of corneal lamellae, peculiar refractive index of corneal lamellae, vascularity of the cornea, relative state of dehydration, swelling pressure of stroma counters the imbibition effect of IOP, and corneal crystallines, water-soluble proteins of the keratocytes. Thank you so much. This session is powered by digital version 2.0, Jyoti Vidyapeeth Women's University. I hope you are satisfied with my digital session. If you have any query, please write in the comment box. I will resolve it. Thank